Morning students, welcome to class 10 biology lesson number 16, cell division 2. In our previous class we have already discussed the mitotic cell division that takes place in animal cells in detail. In this class we will discuss the cell division that takes place in plants and how it is different from animal cells. First, mitosis in plant cells is almost similar to animal cells. That is, most of the phases are all the same, but there is a slight difference in the prophase and the telophase. Let us study that. Interphase. All the three stages, that is G1, S and G2 phase, are the same as in the animal cell. Prophase. It is similar to animal cell except that it does not contain centrosome, hence centrioles are absent. In plant cells, centrioles are absent, so obviously the movement of centrioles towards the opposite poles will not take place. In this stage, asters are not formed like in the animal cell, but spindle fibers are formed. This type of spindle is called anastral spindle. The rest of the changes that take place in prophase, that is the cell membrane disintegrating, nucleolus disintegrating, the chromatin network being converted into chromosomes, all are the same. Metaphase also is similar to animal cells, and in plant cells also the chromosomes align themselves along the equatorial plane. Anaphase is also similar to the animal cell. That is, the chromosomes split into sister chromatids and they start moving towards the opposite poles. Next we come to is telophase. The difference is here in the telophase. Two sets of daughter chromosomes reach the opposite poles. This is what happens at the end of anaphase. Then here they start unwinding and they form the chromatin network. The spindle fibers disappear. The nuclear membrane reappears and so does the nucleolus. It is similar to the animal cell. Now, instead, in the plant cell, you can see that there is a cell wall present. So, obviously, invagination of the membrane or the wall cannot take place. Hence, in the plant cell, a plate is formed. We call it the cell plate. It is laid down by the Golgi bodies and the endoplasmic reticulum. The cell plate is formed at the center and then it moves towards the periphery, resulting in formation of two daughter cells, just like in the animal cell. Now, what is the significance of mitosis? Mitosis is responsible for maintaining the chromosomal number constant in all the cells of the body. Growth. All growth in somatic cells is a result of cell division by this method. Repair of injury is done by division of the adjoining cells in the same way as the growth takes place. Replacement of dead cells by new cells for survival is done by this method. Asexual reproduction in unicellular plants as well as animal is carried out by mitotic cell division. Regeneration of lost parts. Just like in the lizard loses its tail sometimes, it is reformed by division of cells by mitosis. Embryonic development to convert the embryo into an organism is a result of mitosis. Maintenance of cell size. When the cell is fully grown up, it divides by mitosis to form two daughter cells, thus maintaining the cell size. Meiosis. This is the other method of cell division and it takes place only in the gametes, that is, in the sex cells. In females, the ova, and in males, formation of sperms. Meiosis is a type of cell division in which a cell undergoes two successive divisions, producing four daughter cells, each containing half the number of chromosomes as a result as compared to the plant parent cell. Hence, it is also known as reduction division. We have the diploid cells. They divide into two haploid cells and these haploid cells further divide into two, thus giving rise to four haploid cells. Significance of meiosis. Reproduction. Meiosis takes place during the formation of gametes. I told you gametes are formed by a process which is known as meiosis, that is reduction division. Maintenance of chromosomal number constant. In gametes, the chromosome number is halved so that after fertilization, the normal diploid number is restored. Variations. Crossing over during meiosis helps in formation of recombinations or variations in genes or chromosomes. Evolution. The formation of new species from pre-existing ones by modifications in them as a result of variation in their genes and chromosomes is the cause of evolution. So actually, if you go to see, meiosis is the cause of evolution because changes take place because of meiosis. Now we come to a very important part of cell division or cell, and that is the chromosome. Before we study about the chromosome, let us see what this chromosome actually is. Let us see the nucleus of the cell. The nucleus of the cell is made up of 4 parts. The nuclear membrane, which you can see very clearly here in blue. It has pores which are known as the nuclear pores. The nucleoplasm, the purple colored fluid which is present inside this nucleus is known as the nucleoplasm. We have the black dark colored nucleolus and the chromatin network. The thin thread-like structures that you see is the chromatin network. During cell division, these chromatin fibers that are present, these thread-like structures that are present, they condense to form chromosomes. Chroma means color and soma means body. When the scientists were carrying out the experiments and they stained the nucleus, these thread-like structures took the stain very easily. Hence, they called them chromosomes. Chromosomes become visible during the prophase stage of cell division, and they are very prominent during the metaphase. Chromosome consists of exactly 2 rods called sister chromatids. Whether it is a single structure, it is also called a chromosome. The other one which you see having two rod-like structures joined by a centromere, that is also called a chromosome. Any structure for that matter having a single centromere present is called a chromosome. The only difference is this chromosome, we say it is made up of 2 sister chromatids. These rod-like structures are called chromatids. Okay. Structure of the chromosome. Now let us see what is the structure of the chromosome. In each chromatid, that is that single rod-like structure, is present a highly coiled thread of DNA and protein called chromonema. It is called chromonema. Can you see the thread-like structure inside this? It's called chromonema. Each chromonema bear darkly stainable granules called chromomeres. In this, if you see the second one, you can see those dots there. They are called the chromomeres. The chromonema is surrounded by a matrix, and this matrix is covered by a covering which is known as the pellicle. The terminal ends, both the ends of the chromosome, are known as telomeres. Now, let us see its chemical composition. Chromosome consists of 40% DNA and 60% histones. Histones are a type of proteins. The histones and DNA form a kind of complex in which the DNA strands wind around a core of 8 histone molecules. This complex is called a nucleosome. You can see in the diagram that there are 8 histones which are tied up together, and that whole structure is called a nucleosome. Just imagine, you remember when you go to the shopkeeper and ask him for change, he gives you a stack. If you ask 5 rupees change, he gives you a stack of coins which are 5 in number and they are equal to 5 rupees. If you ask 10 rupee change, another stack he gives you with 10 1 rupee coins, and that is the way it is easy to give anybody any change. It doesn't take time in doing so, and obviously it occupies less space. Instead of spreading that whole coins in a dish or any other space, it is better to stack them up that way and keep. Similarly, the nucleus is a very small structure, and in that you can imagine the DNA thread that is present, the double helical DNA that is present, it is almost 2 meters in length. And there are histone molecules also. So all of them, keeping it in a very small space is very difficult. Hence, they are tied up like this way to form nucleosomes and then stored. During condensation, what happens is they wrap around more tightly and they are packed together to form structures that which we call chromosomes. You can see in the diagram, they are the chromatids or chromatin fibers. If you open them up, they look like this way, okay, with the DNA and the histones. A single chromosome may have 1,000,000 nucleosomes. So you can imagine. Structure of the DNA. Now the question arises, what is this DNA? DNA is deoxyribonucleic acid. It is a macromolecule. It consists of 2 strands. You can see those 2 strands coiled around a common axis. That dotted line is the common axis in a double helix, giving it an appearance of a spiral staircase. Each DNA strand is composed of repeating nucleotides, which are made up of 3 components. The nucleotides have 3 components: phosphate, pentose sugar, and which are arranged in the lengthwise. That means the edges that you see, it is around that area, and the nitrogenous base attached to the sugar inwards. You can see the nitrogenous bases in the forming that ladder-like structure. That is the nitrogenous base. It is there. Okay. The nitrogenous base joins the complementary nitrogenous base of the other strand by hydrogen bonds. We will go into this in detail in the next slide. Structure of DNA, now you can see it very clearly here. These 2 strands come together like a ladder-like arrangement with the nitrogenous bases forming the rungs of the ladder. The 2 side rows are the straight lines, and the center you have the rungs of the ladder. There are 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. Adenine pairs with thymine always, forming a double hydrogen bond. Adenine will never combine or never form a pair with guanine or cytosine. It only forms a pair with thymine. And so too, thymine always forms a pair with adenine only. Guanine pairs with cytosine, and there are 3 hydrogen bonds in between them. Again, cytosine will only be with guanine and guanine only with cytosine. Now let us come to what is genes. Genes are the hereditary units which carry a particular trait from one generation to another. Genes are specific parts of the chromosome, and they are composed of DNA. They determine the hereditary characters. They are responsible for transmission of characters from one generation to another. They always occupy a fixed position on the chromosome. If a particular sequence of genes are present as 1, 2, 3 in my chromosome, then in yours also, and that particular chromosome, the position would be 1, 2, 3 only. Okay. They are arranged in a single linear order in the chromosome. Beta, that is all about the cell division, chromosome structure, and genes. With this, we end this chapter.